Hello everyone, so let's start with the part 3 of the amino acid metabolism. So here we are going to discuss about the decarboxylation process, the enzyme responsible for the decarboxylation process, and what will be the fate of the ammonia after the process of decarboxylation, where it is going to enter, where will be the consequences of the urea or formation of the urea in your body. This all we are going to discuss with or in this particular slide. So let's start with the decarboxylation. So basically you see the decarboxylation of amino acids. So what do you mean by the decarboxylation? Decarboxylation means the removal of a carbon dioxide molecule. So basically if you see decarboxylation, it is the removal of a carbon dioxide molecule from the amino acid with the formation of amines. So if we take the basic structure of the amino acid, so this is the basic structure of the amino acid. See here, I have removed one carbon dioxide and finally it has formed one amine molecule. So this is the basic strategy of the decarboxylation reaction of the amino acid. Now you should know that this amine, whatever it has formed, it have different physiological activity in the human body, which include different enzymes which are found in the form of the amines, as well as different neurotransmitter, and the enzyme which is responsible for the process of decarboxylation. It is nothing but the decarboxylase enzyme, and there is always a coenzyme which is involved in this process. It is known as the pyridoxal phosphate. Now you see, in the decarboxylation of the amino acid, basically how it takes place, we have divided it into different types. First of all, if we consider the alpha decarboxylation, the alpha carbon means the carboxyl group which is associated with the alpha carbon, it is removed. You can see here, this is the alpha decarboxylation of the amino acid, right? Then we have the omega decarboxylation. So here you can find in the omega decarboxylation the carbonyl group, it has been removed from the omega position, right? Then apart from that, you have other reaction which includes the decarboxylation with transamination. In this process, both the step of the decarboxylation as well as the transamination, it takes place simultaneously. For example, you can see here, so here this is the amino acid, right? So here you can see here this amino group, I have already told you the transamination. This amino group will be transferred to this position, as well as this carbonyl group, this CO2 molecule will be removed, resulting in the formation of this particular product, right? Apart from that, we have one more type. It is the decarboxylation with conjugation of two molecules. So here two molecules, it get conjugated along with the liberation of a carbon dioxide molecule. So in all this process, you can see there is a liberation of carbon dioxide molecule, which signifies the process of decarboxylation from the amino acids, right? Now why it is important? The first question, why it is important? So you should know that the significance of amino acid decarboxylation. There are different chemical messenger in our body which uses these amino acids after their decarboxylation. For example, we are going to take some classic example of amino acid which are involved. So first of all, formation of physiologically active compound. Here you can see from the glutamate, if we decarboxylate the glutamate, it forms generally the gamma amino butyric acid. So it is a neurotransmitter which is present in the CNS. We all know that, right? Similarly, from the histidine, if we decarboxylate a histidine, it forms the histamine. So what is the significance of histamine? We all know that histamine is a mediator of inflammation, it is a mediator of allergic reaction, it is a mediator of pain. So all these things are actually responsible for various physiological processes in the human body. So regarding the histamine, you can say that a lots of the histamine you can find in the inflammatory places, right? So you can say, or you can predict that the histamine is a signaling molecule for the inflammatory response, right? Apart from that, this histamine, they act as a vasodilator, right? It dilates the blood vessels. Moreover, it mediates the inflammation and it mediates the pain in the body, and it is responsible for the allergic reaction. So all the allergic reaction, mostly they are caused by the histamine. That's why for the treatment of the allergic reaction, we used to take antihistaminic drug like cetirizine and all, right? Moreover, this histamine, it stimulates the HCl secretion in the stomach also, right? Now, apart from this histamine, there are some other types of the active compounds, physiologically active compound, which are actually formed by the decarboxylation of amino acids. Let's start with few more. The next one regarding this amino acid product which has formed from the decarboxylation, it is the tryptophan. We all know that it is the serotonin. We all know that the serotonin has formed from the tryptophan, right? The tryptophan, once it will undergo decarboxylation, it will form the serotonin, right? This serotonin, they act as a vasoconstrictor in our body, we know that. Moreover, this serotonin, it also regulate the arterial pressure, the body temperature, the respiration, kidney filtration, and many more functions in the nervous system actually, right? The next one, it is the dopamine. So this dopamine, it actually formed from the tyrosine. Tyrosine is an amino acid. The tyrosine, once it undergoes the decarboxylation reaction, it forms the dopamine. So what is the role of the dopamine? Actually, the role of the dopamine in the CNS is that it is a precursor of epinephrine and norepinephrine. Moreover, it is a mediator of the central nervous system, right? The next one, it is your GABA. That is your gamma amino butyrate. So it is formed from the glutamate. Now what is the role of the GABA? It is an inhibitor mediator of central nervous system. Moreover, it is used as an anticonvulsant drug, right? Now we are moving to the next slide. Now regarding the transport of the ammonia to the liver, now we all have come towards one thing, that these amino acid, once they are subjected for transamination or decarboxylation, in the decarboxylation, a carbon dioxide molecule has formed. In the transamination, the amino acid has been transferred in the different groups, right? Now what is the fate of this ammonia, or in the during the time of the deamination, a amino group, it is liberated, right? Now what will be the fate of this ammonia? Now this ammonia, it will be transported to the liver. So basically we have two mechanisms for transport of the ammonia. The first mechanism, it is the glutamine synthesis. So here the glutamine synthase enzyme, it will combine with the glutamate, right? Now in the presence of the glutamine synthetase, this glutamate, it will combine with the ammonia, right? This is the first mechanism. Moreover, we have one more mechanism where alanine will take part. So here in the second mechanism, it generally involves in the muscle cells. So it involves the transamination of the pyruvate to alanine, right? So these are the two processes which are basically take part in the human body. So let's start with the first one, the glutamine and the glutamate. So basically you see the glutamate and the glutamine relationship. What is the relationship between these two? The ammonia nitrogen. I repeat, ammonia nitrogen means the nitrogen of the ammonia. NH3 is the ammonia, right? It can be transported as a glutamine in the human body, first of all, right? Moreover, this is the first line of defense in the brain cell. This brain cell, it removes the ammonia with the help of this particular mechanism, right? Now this glutamine synthase, it catalyzes the synthesis of the glutamine from the glutamate and the ammonium ion in an ATP dependent reaction. This glutamine synthase catalyzes the synthesis of glutamine. So you need this glutamine synthase enzyme, right, for the synthesis of the glutamine from the glutamate. Means glutamate, it is already present in your body, so now it will take up the ammonia to form the glutamine, and this process, it is catalyzed by the enzyme glutamine synthase, right? Now the nitrogen of the glutamine can be converted into the urea in the liver by the action of the glutaminase, which is found in the liver. So next enzyme, it is the glutaminase. So basically you need the glutamine synthase and then the glutaminase, right? Now the hydrolytic release of this amide nitrogen from the glutamine as a ammonia, it is catalyzed by glutaminase, favors glutamate formation. So I'll go in the details in the next slide, where I'm going to give you the methods, the steps, how it get converted. Moreover, the concentrated action, or you can say the concerted action of the glutamine synthase or glutaminase that catalyzes the interconversion of the free ammonia ion and glutamine. So basically you see here, this is the glutamate molecule, here this is the ammonium ion, right? We already discussed about the glutamate synthase. So this is the glutamate synthase. It is going to act here. We already told you it is a ATP dependent reaction, so ATP will involve, and this is the glutamine, the formation of the glutamine. Now this ammonium ion, it has entered into the glutamate. It will form the glutamine, and along with it will form, and this glutamine actually it will form the glutamate here, right? Next, the glucose-alanine cycle and the role of glutamine, the next part, which actually takes place in the muscle cells, right? The first of all, you should know that the transport of the amino acid, it also takes place in the form of alanine, as I have already told you, right? Now the nitrogen here, it is transported from the muscle to the liver in two principal transport forms. Now this nitrogen will be transported from the muscle to the liver, right? And there are two transport forms. So first of all, you see here the glutamate is formed by the transamination reaction. We have already come to know that, right? But the nitrogen is then transferred to the pyruvate. I repeat this line once again. The glutamate is formed by the transamination reaction. We have already covered up in our previous slide, right? Now this nitrogen is then again transferred to the pyruvate to form alanine, which is released in the blood. Now this alanine will be released in the blood. From the glutamate, it will form the pyruvate. Again it will form the alanine, right? Now the liver will take up the alanine from the blood and it will convert it back to the pyruvate by the process of transamination. And this pyruvate can be used for the gluconeogenesis, means for the genesis of the new molecules of glucose, and the amino group eventually appears as the urea. And this transport, it is referred as, this method, it is referred as the alanine cycle. Now if we continue this thing, so you see here, this is the process in the muscle cells, which I have already told you regarding the conversion. And this is the way how, see, this is the muscle cells, right? From here, what is the conversion? We have given you a stepwise conversion of this process, right? So this is the two basic mechanism of their conversion, of their transport. Now let's start with the ammonia intoxication. We are telling that the ammonia has formed. Now you should know that this ammonia, it is toxic to our body. We have a minimum amount of ammonia threshold in our body. Means once this threshold it is crossed, once the ammonia level has crossed in our blood, it will show different toxic reaction in our body. So without wasting time, let's start. First of all, the ammonia, it is produced in our body by the enteric bacteria, first of all, right? Apart from that, it is absorbed in the portal venous blood, and this ammonia, it is also produced by the tissue that are rapidly removed from the circulation by the liver and converted into the urea. So this ammonia, whatever it has formed from the bacteria or from the body cell, it is converted into the urea by the liver. Now this urea has to be moved out from the body because it is toxic in nature, right? Now the normal amount, it is generally 10 to 20 gram per deciliter. It is the threshold value of the urea. It should not cross this value in our blood, right? Now you should know that this process is very essential. Ammonia intoxication process is very essential, since the ammonia, it is toxic to our central nervous system. This ammonia can lead to the destruction of the central nervous system, right? Now, so should the portal blood bypass the liver, or systemic ammonia levels may rise to the toxic level. I repeat this line. So if this portal blood containing the alanine, which is having the ammonia, right, if that bypass the liver, then the level of the ammonia in the blood, it rises the toxic level. So in a patient with the liver disease, the toxicity of the ammonia, the chances of toxicity with the ammonia, it rises actually. So this occurs in severely impaired hepatic functions or development collateral links between the portal and the systemic veins in the cirrhosis. So both impaired hepatic function as well as cirrhosis. Here the liver function, it is very less. In these cases, the ammonia intoxication takes place in the human body. So ammonia intoxication, if I repeat, this excess of the ammonia, actually it depletes the glutamate. We know that this ammonia, it will combine with the glutamate to form the glutamine, right? And hence the level of the GABA in the brain, it will come down, because we know that this glutamate, it is used for the synthesis of the GABA. Now if the high amount of the ammonia is present in the blood, this ammonia will be used by the glutamate to form the glutamine, and resultant effect is that low amount of the GABA will be present in the brain, right? Moreover, to compensate the glutamate, there is another function in our body. So what we used to do, the alpha ketoglutarate is used, and the decreased concentration of which subsequently depresses the TCA. TCA is the tricarboxylic acid reaction, so it depresses the TCA and thus deprives the brain cells of energy. Now this excess of the glutamine, it is exchanged. The excess of the glutamine in the human blood, in the brain, it is exchanged with the tryptophan, and we all know that the tryptophan, it is a precursor of serotonin, thus resulting in the hyperexcitation. Thus you can relate one thing: if there is a high amount of the ammonia in the blood or in the brain, right, this will deprive the glutamate in a general sense, because this glutamate will combine the ammonia to form the glutamine, and it will degrade the level, or it will degrade the amount of the GABA in the brain. Now we have another function or another mechanism where actually this glutamate can be replaced with the tryptophan. Now if tryptophan is replaced, then what will happen? Tryptophan is a precursor of serotonin. The amount of the serotonin in the human body or in the human brain, it will come down, and we all know that the serotonin, depletion of serotonin may result in the hyper excitation, or the person, he may suffer from anxiety. Thus the symptom of ammonia intoxication in the human, it includes the tremor, slurred speech, blurred vision, coma, and sometimes it can lead to the death also. Now let's go for the urea, or you can say this is the ornithine cycle. So what is the function of this cycle? First of all, you should know that this urea cycle, it detoxify the liver. It proceeds with the liver. So detoxification pathway, it is processed only in the liver, first of all, right? The next thing is that it is localized in the cell, basically in the mitochondria, and sometimes this also occurs in the cytoplasm. And the enzyme in the mitochondria responsible for this process, it is the carbamoyl phosphate synthase 1. This is the enzyme in the mitochondria which is showing an active function in the urea cycle, and it can acidify an organism, right, as it consumes the bicarbonate ion. This particular process, and needs this process, this urea cycle, it needs 3 molecules of ATP, but 4 energy rich bond are need to be formed here, right? Now it's connected with the citrate cycle through the fumarate. I will show you how it is connected in the next slide. Moreover, urea is the end product of nitrogen metabolism, and this urea, it comes out from the body in the form of urine, right? So detoxification of the ammonia in the liver. So this is the process. So let's discuss here. So this is the cytoplasm, this is the mitochondria. You can see here the process. So we start with the alanine. This alanine has come from where? It has come from the muscles, right? Now this alanine can form as a glutamate. Similarly here, this undergo transamination to ketoglutarate to form this amino acid to form glutamate. Now it will enter here and it will enter here as a glutamate, right? With the help of this particular reaction, you can see here the glutamate dehydrogenase, here it will act, it will release the ammonium ion, and finally this enzyme, particularly the carbamoyl phosphate 1 synthase, it will lead to the formation of this carbamoyl phosphate, right? Now similarly, these are the other product, this conversion of glutamate to aspartate and oxaloacetate. Now you concentrate here. Now this carbamoyl phosphate, it can, first of all, the first step, it is a conversion into the citrulline. This is the first step, right? Now this citrulline, it will come out, right? This citrulline, it will utilize this ATP and it will form the aspartate. From this again, it will form the arginosuccinate. It will leave 1 molecule of fumarate, and this arginosuccinate will now form the arginine. These processes are taking place in the cytoplasm, I remind you, right? It will form the arginine, and from here you will get the urea. Again, once it will leave out 1 molecule of urea, it will again form the ornithine, and this ornithine will again enter the mitochondria. So this is the process. The basic things are here: the carbamoyl phosphate synthase 1. This is the carbamoyl phosphate synthase 1. The next one is the arginosuccinate synthase, right? and succinate lyase and arginase. So these are the enzymes which are responsible. So this is the basic method. So here this is the cytoplasm, this is the mitochondria. See, you can see here, you can demarcate where the process it takes place. I repeat once again, this ammonia, this is the alanine, it is coming into the cytoplasm. It will get first converted into glutamate, then again this glutamate will enter the mitochondria. From here it may get converted into alpha ketoglutarate, right? Or sometimes with the help of the enzyme aspartate, aminotransferase, it can get converted into aspartate, right? This glutamate, again with the help of the enzyme, the glutamate dehydrogenase, it will liberate the ammonia. This ammonia will, with the help of the carbonyl phosphate synthase 1, it will form the carbonyl phosphate. Again the citrulline, from the citrulline it will form aspartate, arginosuccinate. Then the fumarate molecule will be left out, and then next it is the arginine, and it will leave out the urea, and finally it will form the ornithine. So this is the basic cycle actually of the method, or you can say this is the basic cycle of detoxification of ammonia in the liver, right? Now interconnection of the urea cycle with the citrate cycle. So what is the interconnection? Tricarboxylic acid TCA cycle, right? So this is the interconnection. So here you can see this is the urea cycle, which we have already discussed in our previous slide. So what actually happened here in the urea cycle? If we start the citrulline, it get arginosuccinate, then arginine, right? It will liberate the urea, it will form the ornithine, it will enter the process, right? This is the cytosol actually. This ornithine will enter again. Here we will get the carbonyl phosphate, and this is the mitochondrial matrix actually. This part, it is a mitochondrial matrix, and similarly in the other cycle which is associated with this, that is the TCA cycle. Here you can see the citric acid cycle. Here you can see this arginosuccinate, it can form the fumarate. The fumarate will get converted into malate, into oxaloacetate. As we all know that we have already studied this cycle. So this fumarate, it will get converted into malate, into oxaloacetate. This is the cycle. So the conjugation means, or you can say the relationship of these two cycles, it occurs basically at this particular step, that is the arginosuccinate. Here, from here the aspartate will get converted into arginosuccinate, and similarly this citrulline get converted into arginosuccinate. So it will lead to the formation of the fumarate in the TCA cycle, and it will lead to the formation of the arginine in the urea cycle. So this is the relationship actually between these two cycles. So now regulation of the urea cycle. Who is going to regulate this urea cycle? So first of all, you should know that the mitochondrial carbonyl phosphate synthase 1, it is also known as CPS1. It is an enzyme which will regulate the urea cycle. This is a fast factor. Moreover, this CPS1, it catalyzes the first connected, or you can say it catalyzes the first committed step of the urea cycle, as we have already discussed in the previous slide, right? Now this CPS1, it is also an allosteric enzyme sensitive to the activation by N-acetyl glutamate, which is derived from the glutamate and acetyl coenzyme A. So we have already discussed about the allosteric enzyme in our part 2. So CPS1, it is also a allosteric enzyme, and it is generally sensitive to the activation by the N-acetyl glutamate, right? So thank you everyone for your patience hearing. If you like the video, you can press this like icon. You can share this video with your classmates and colleagues, and for further notification from this channel, you can press the bell icon. Thank you once again.